This is our lab pet, Jerry. While some labs have rats or mice, we have a giant single-celled amoeba. And while it may not look like Jerry is doing much, if we speed the footage up a bit, we see that Jerry is actually extremely mobile. It looks more like a Venom symbiote than pretty much anything else.
Eddie? What was that?
Oh, I have a parasite. Yeah. Name is Jen.
Jerry is a slime mold whose scientific name is Physarum polycephalum, and normally Jerry can be found roaming the forests and countrysides of most temperate places on Earth. Jerry eats both decomposing plants as well as various microbes. But don't let the word mold fool you. Jerry is not a fungus, and unlike a fungus, Jerry really does roam. Like other amoeba, Jerry crawls along looking for food, building up biomass and taking it along for the ride. This is very different from a fungus that stays in one place, sending out long hyphae that grow into massive networks. Because of this, Jerry has a huge suite of sensory capability to find food and protect itself that we'll be putting to the test today. And while Jerry is one gigantic cell, that doesn't mean that Jerry isn't intelligent. Oh no, in fact, Jerry is a genius and puts its full sensory suite to very good use. Let me show you what I mean. This is a little 3D printed maze, and after sterilizing it with alcohol, I'm going to fill the bottom with some plain agar. This will give Jerry a moist surface to crawl around on without drying out. Then we'll put some oats at the start and end of the maze, and a little sample of Jerry at the start. We also put a single oat at the midway point as a little snack for Jerry. The nice thing about Jerry being one giant cell is that you can literally just scoop up a little bit of goo flesh and plop it onto some oats, and it'll reconstitute itself into a single cohesive unit again. After initially building up enough biomass, Jerry sets out to explore, and beelines for the halfway point where the oat snack is waiting. After pausing for a quick bite, Jerry proceeds to sprint to the end of the maze. Now, to be fair, this all looks much faster than it really happens. This whole time-lapse was taken over the course of 2 days, so while Jerry has remarkable amounts of locomotion for a giant single cell, you're not going to have to worry about Jerry running you down. Yet. Today, we're going to be learning all about Jerry, what they are, how they do what they do, and how I learned to stop worrying and love Jerry. And near the end, we're going to actually try reading Jerry's mind. So there's lots to do, let's jump right in. While it's very cool to watch Jerry on these super long time lapses, let's take a moment to zoom in a little and see the inner machinery of Jerry. Up close, the first thing that you'll notice is the flow. Jerry is not solid at all, and is a network of tubes and pumping fluid. And you'll notice that the fluid will actually change directions. This is an active process of Jerry redistributing nutrients and biomass to where it's needed. What's incredibly weird is that Jerry uses the same proteins that our muscles use for moving around. These proteins, actin and myosin, are basically like interlocking combs. When given a chemical signal, myosin will start to aggressively climb up actin, causing the cell to contract. At least that's how it works in muscles. But watch what happens when I speed this clip of Jerry up a little bit. Look at how everything pulsates. This is the result of the network of actin and myosin that Jerry builds and controls. Jerry also makes extensive use of kinesins and other motor proteins. If you've never seen these before, they're the super cute proteins that walk up and down tubulin fibers inside of cells. So technically, Jerry does have legs, but only on the inside. Jerry also has proteins that allow specific areas of Jerry's body to stiffen on command. By controlling which areas stiffen or relax and which are pumping fluid, Jerry can push the internal fluid where it's needed and crawl along the ground. For example, if we look at a growing edge, we can see that it actively pushes the internal goo into and out of the area. And all the flow means that any nutrients that get absorbed are quickly distributed to the rest of the body. And we'll see some experiments in a moment where we can actually see that distribution happen. Since there's no inherent front or back to Jerry, it can actually investigate multiple avenues at the same time and control all the goo squishing with remarkable amounts of finesse. Even making repairs is done with incredible speed. If I take a needle and carefully puncture one of the big goo arteries, as expected, a bunch of goo comes out. But you'll notice that it's not really a lot of goo, as the flow halted very quickly. But then to our amazement, we watched over the course of a few minutes, all the goo just gets sucked right back in, the channel is repaired, and the flow of the network has restarted. Let's do that again because it was really cool to watch. It is amazing how quickly it just slurps the goop back in and fixes the leak, all while the rest of its body is still going about its business, eating oats and generally vibing out like it's no big deal. Now, watching this, you might accidentally think that all the little round things flowing around are some sort of blood cell, but they're not. As I've said, Jerry is just one giant cell. These are mostly nuclei, the DNA storage and processing center of cells, and the goo is cytoplasm, not blood. Normally, eukaryotic cells have one nucleus, though there are some exceptions like muscle cells where you get a few extra to help deal with the intense amount of protein that needs to be constantly produced. But Jerry is all nucleus all the time. Instead of dividing itself up into individual compartments, the nuclei just coexist in one giant mass of cytoplasm. In fact, Jerry has the highest cytoplasmic flow rate ever found in an organism. One perk of this way of life is that if you break a piece of Jerry off, there's always at least a few nuclei left that can keep up the basic cellular functions needed to live. For us, this is nice because it makes Jerry very easy to propagate. Though it's one thing to say that these are nuclei and another thing to just, check. So I'm going to use a special dye called Hochest 33342. Yeah, that's fun to say. It's a live cell stain, which means it won't harm Jerry and can be applied without having to fix the tissue first, though calling any part of Jerry tissue feels weird. Anyway, Hochest stains DNA fluorescent bright blue, so if we switch to the blue channel and turn on the fluorescent light system on our microscope, we can have a look. I had to take a long exposure as the glow was kinda dim, but sure enough, we see lots of blue nuclei embedded in the non-moving goo, and the veins are glowing blue as the nuclei streak past. I combined a white light and fluorescent image to make this view. We see tons of nuclei flowing in the veins, but are especially concentrated near the growing edge. Though while we're doing this, I have another dye I want to try. In a previous video, we used a dye called Rhodamine 123 to visualize the mitochondria inside skin cells that we had grown. Mitochondria are some of the most important organelles in a eukaryotic cell, producing most of the energy a cell uses. Say it with me now. Roses are red, it's hot like hell. Mitochondria are the powerhouse of the cell. Anyway, they're basically bacteria that many billions of years ago got subsumed by one of our cellular ancestors. But instead of being digested, the two cells ended up in a symbiotic relationship. And to this day, they're weird, wiggly, worm-looking things and maintain their own small genome. So, what's the mitochondria situation inside Jerry? Something as mobile as Jerry is going to need a lot of energy, so as expected, we see a lot of mitochondria. But they're small and dense instead of being the long noodles that we get in human cells. Which, I suppose that makes sense. It's an organism that exists in a constant state of flow. You don't really want things clogging up your pipes. Though goo flesh and cytoplasm aren't the only things that flow. One thing that's faster than a hungry Jerry is the endless torrent of news we're inundated with every day. Figuring out what's true and what's just the opinion of a billionaire that bought a news outlet is getting increasingly difficult, and it's so easy to fall into our own little echo chambers and miss really important stories, or believe a story just because we like the sound of it, even if it's not factually accurate. Thankfully, there is a solution to this, and that's the fantastic sponsor of this video, Ground News. Ground News is a news aggregator that collects all the major stories being reported and allows you to compare how different news outlets are reporting the same thing, if they have a political bias, see who owns those outlets, and provides a factuality rating so it's easier to see how reliable an outlet's reporting tends to be. There are also a lot of stories that slip through the cracks because they don't fit the political bias of a particular news outlet. Ground News shows you these underreported stories from either side in a dedicated feed, so now you can find out if you've missed an important story because your usual outlet didn't think it would fit their audience. Here's an example. If we look at the coverage of the recent American withdrawal from the World Health Organization, left-leaning networks talk about the devastation this will cause and how doctors are extremely worried. More centrist networks talk about the impact this will have on fighting disease in the developing nations and the spread of TB or HIV. Whereas on the right, you have articles claiming that this was a genius move because freedom or something. But with Ground News, that's just not a problem anymore. You can see what everyone is saying, and the factuality rating helps you make sure you take things with a grain of salt as needed. So if you want to upgrade your news experience and enjoy a more truthful feed, head on over to ground.news/emporium to get 40% off the unlimited access Vantage plan, or scan this QR code. But now, back to Jerry. Alright, let's zoom back out again and stop staring at Jerry's junk. Instead, let's take Jerry for a walk. I'm sure that there's one question that's been weighing heavily on your mind, and that is, of course, what's Jerry's favorite flavor? Does Jerry like spicy or prefer sweet? And is flesh on the menu? Let's find out. We set up a petri dish with 5 oats that have each been flavored. We've got sweet, spicy, salty, sour, and umami. So let's see if Jerry has a preference. It seems as though Jerry has a taste for heat because the first oat consumed was the spicy one, so capsaicin evidently doesn't bother Jerry at all. From there, Jerry splits and tries both the sweet and umami oats. Jerry seemed to take a lot of time with the sweet one, but ate the umami one very quickly. Maybe Jerry doesn't have a sweet tooth, or just wanted to enjoy the sweet one for longer. Now for the really interesting bit. Jerry heads towards the sour oat. This time, it seems like Jerry just went right past it and showed no interest in eating it. And this was quickly followed by the salty oat, which also seemed unpalatable. Both oats initially were untouched, though it seems like after licking the salt off the oat, it was eventually made palatable enough to be eaten, though the sour oat was never touched. We'll have to do more testing, but it seems like salty humans aren't on Jerry's menu. And I guess if you're worried, douse yourself in lemon juice. It probably won't help, but the burning sensation will take your mind off it. But more to the point, Jerry is an entirely harmless organism to humans. I realize that this whole video up to this point has been giving mad Last of Us vibes, but not only is Jerry completely non-pathogenic, Jerry is technically edible. Though the reports are that Jerry tastes awful, which does make sense for an organism nicknamed the dog vomit slime mold. And frankly, I can't be asked to repeat that particular experiment as I draw the line at tasting Egyptian mummy. Though, at least as microbes go, Jerry doesn't smell bad. So, that's nice at least. I know it may look like Jerry would smell foul, but Jerry sort of smells like decaying leaves or forest dirt. Of all the things we've grown in the lab, Jerry is by far the least off-putting smell-wise. And maybe in the future we'll genetically modify Jerry to smell really nice. Now, the one problem with the taste test was that we can't see the nutrients as they distribute through Jerry's body. So let's try a similar test, but instead of a flavor, we're going to use a dye to act as a tracer and see how it distributes. What's amazing about this is that if we zoom in a bit, we can actually see how fast the dye moves through Jerry. It is much faster than the passive diffusion of the dye through the agar. We can see that faraway oats take on a pink tinge almost immediately after Jerry makes contact with the dye. But then we see Jerry's whole body take on an orange hue. Let's try this again with more colors. We wondered what would happen if we used different colored dyes, and if we would see Jerry change colors as the different dyes mix in their body. This time we used 3 separate pieces of Jerry to start with, so on top of showing how nutrients distribute, this is also a great demonstration of what happens when identical bits of Jerry meet. They just fuse together with no real fuss, and within moments have adjusted their tubes to make sure everything is flowing properly. With the dyes, we see Jerry go from pink and blue to a really fun shade of green. So not only is Jerry a great pet, but you can accessorize your Jerry too. I suspect with the right mix of colors we could dress Jerry up as Venom for Halloween. Alright, one last time, we're going to try this again, but this time we're using a UV light. The dyes we chose glow under UV, so with an orange filter on the camera to remove the purple light, we can see the dyes glowing brightly. This way, we'll see the exact moment the nutrients start being uptaken, as the glow allows us to see far lower concentrations of dye. To my delight, this worked even better than expected, and you can actually see just how far and fast Jerry moves the dye once it starts being consumed. Eventually, the entire plate is a glowing network of tubes, and the oats are not but glowing husks. Now, one of Jerry's most amazing abilities is their incredible path optimization. When given a plate of food sources in many locations, Jerry finds them and builds a network of interconnected tubes to efficiently distribute the nutrients as we've seen. But it's not just their ability to circulate the nutrients that's amazing. Even just where the tubes end up is a matter of extremely efficient positioning. Famously, this ability was once used to see how Jerry compares to humans by a Japanese group. Using a large agar plate, they put a map of Tokyo underneath, and then placed oats on the agar in corresponding positions that the Tokyo subway stops are arranged across the city. The idea was to see how Jerry would have connected the stops had they been in charge. To make it more accurate, they used a projector to shine light onto the plate, such that the intensity of the light mirrored the geographic topography. Jerry hates light, so this helped discourage Jerry from growing in areas where it would have been bad to build a train line. Once they inoculated the plate, all they had to do was wait. After a day or two, when the whole plate was covered with Jerry, the main branches that had been formed between the oats weren't too dissimilar from the arrangement of the actual train lines. Sure, it wasn't perfect, but it wasn't bad for a thing that doesn't have a brain, let alone any cells. For our replication, we didn't end up using the projector, but you can still see that Jerry does a great job interconnecting each of the oats efficiently. We're going to get some larger petri dishes and maybe build a projector setup so we can try this again in a future video. Jerry is so good at problem solving that we began to wonder what was really going on under the hood. Jerry may not have cells, but that doesn't mean Jerry couldn't be thinking. It would just have to be a different kind of thinking than we're used to. Because, again, no cells. So you can't have action potentials coming from discrete units, but perhaps there's still electrical activity helping to direct the whole mass. So we figured this would be the perfect opportunity to fire up the system we had on loan from Multi-Channel Systems. Over the past few years, we've been working on building a system to play video games with living human neurons, with the goal of eventually using it to play Doom. In our last episode of the series, Multi-Channel Systems lent us this amazing system to allow us to take our first recordings of the neurons that we've been growing. We went over the setup of the system in detail last time, but here's the TLDR. The system connects together in 3 pieces and connects to my laptop via USB cable. The head stage is this clamshell piece, and it's where you put multi-electrode arrays with your cells on it. Multi-Channel Systems also was kind enough to send us a few of their incredible electrode arrays. Since these are meant for recording human neurons, the electrodes are all incredibly tiny and are clustered into a small area for maximum neuron resolution. Thankfully, they sent us a few spare, so we haven't used all of them for neurons yet. This means we could try one with Jerry. And of course, we have our homemade neuron arrays, which we tested in that video as well. Unlike the fancy multi-channel system ones, these are made from cheap PCB material stuck onto a glass backplate, but we showed in the last video that they work great for recording from neurons. Unlike the commercial electrodes, ours are laid out specifically eventually to play a video game with neurons. As such, the electrodes are larger and spaced much further apart, so it'll be interesting to see how Jerry behaves on each array. One nice part of working with Jerry is that I can just take a blob of oats and Jerry and slap it onto the arrays, rather than having to carefully tend and grow the neurons like usual. And insofar as Jerry is used to growing on the forest floor, Jerry doesn't mind a little bacteria, so for short experiments, we don't even need to worry about intense sterile technique as much. Anyway, just like the last video, we slot our array with Jerry in it into the head stage, then close it up and wrap it with aluminum foil to act as a Faraday cage. We learned this was necessary last time because of the intense amount of electrical interference in our lab. This also happens to bonus as a light shield, which will keep Jerry happier about the whole situation. Starting off, this is the program interface for controlling the system and taking recordings. Starting first with the array for multi-channel systems with a blob of Jerry on it. Right off the bat, the first thing to notice is that on average, the electrical activity is lower voltage than neurons. And that kind of makes sense, since it's easier for discrete units of a cell to build up and release significant pulses. But we do see distinct spiking activity where the voltage swings rapidly. As I said earlier, the electrodes on this array are all very close together, so they only cover a very tiny patch of Jerry. As such, the signals we're seeing on each channel are very similar, but let's see what happens if we stimulate some of the electrodes with a voltage pulse. Keep in mind, the system will automatically remove the stimulation pulse from the recorded signal, so any deviation we're seeing is electrical activity by Jerry. When we fire the stimulation pulse, we see that suddenly the stimulated electrodes show a massive positive spike in voltage, which then swings negative before slowly coasting back up to normal levels. But the electrodes that weren't stimulated still show a reaction, and firing the stimulator multiple times can push the negative part of the pulse deeper. This looks like a fairly classical action potential, albeit a bit slow, so maybe slightly more like a muscle firing than a brain, but still. Let's try this again with our homemade electrode array so we can examine a wide area of Jerry and see how that looks. Right off the bat, this looks way different than the high-density array. Now we can see larger voltage spikes and swings, presumably because now we've got a better view and have the growing edge of Jerry sat on top of some of the electrodes. When we try the stimulator again, we see a massive spike on the various electrodes, but this time the far away electrodes show some really neat behavior. As you get further away from the stimulator, we see a pulse, but it's inverted. I wonder if what we're seeing is Jerry recoiling away from where the pulse originates and pushing mass to further away areas. Honestly, seeing this immediately made me think that we could train Jerry to play video games just like the neurons. Sure, it's going to need a different layout of the array and probably a different control scheme, but Jerry is so electrically active that with the right setup, I think we could build a Jerry-based control loop. But, I mean, that's going to have to be for another day. Honestly, Jerry is such an amazing organism that we could keep doing demos and experiments like this pretty much endlessly. But this video is getting pretty long and Jerry needs a nap. So we're going to be back to teach Jerry more tricks and see even more weird and wonderful things Jerry can do in future videos. So be sure to subscribe so you don't miss it. I think it's safe to say that Jerry is a pretty amazing organism, and to show our love of Jerry, we've just added this amazing new Jerry design to our store. Like all of our amazing designs, it's available on super comfy apparel, mugs, mouse pads, and more. And to show our love of Jerry even more, use the coupon code GOOLIFE for 10% off your order. While you're there, be sure to check out our handmade posters and other designs, and of course, sign up for our newsletter. We're working on building a Jerry kit and getting our store ready to sell DNA, bacteria, and other cool organisms. So you'll be the first to know when we release them. And of course, I have to take a second to thank our amazing patrons and channel members that make these videos possible. Patrons and members also get access to our supporter Discord, so if that's something you're interested in, or just want to help keep the flow of science videos coming, there's some links below. That's all for now, and we'll see you next time.